We're going to begin the explanation of exactly how the DNA molecules break and rejoin to allow for gene rearrangement. The mechanism is quite complicated, and the first thing we're going to look at is the structure of the DNA signals, that is, the nucleotide sequences that make this whole thing possible. So here is an old illustration of the three different genes, and I warned you that each one of these sections has probably more complexity than I bothered to illustrate previously. So let's take a portion of this and show where you find these particular sequences, the recombination signal sequences. And so here is just a part of the lambda, the kappa, and the heavy chain. And if you'll notice quickly, you're not going to see any funky stripe stuff around the constant regions. And that's also true of the constant regions that we haven't shown on the heavy chain or the lambda light chain. So you can see from this, though, that every variable region is going to have a downstream or 3' signal sequence. They're not all identical, but they're similar. All of the J's will have an upstream version of some kind. And the D region that I'm going to join in between them will actually have things both up and down. Now, the interesting thing about this is it looks like, if you look at the D region, that the recombination signal sequences take up more space, or they're longer than the actual D region itself. Well, while this may exaggerate to some degree the relative sizes of these signal sequences, it really doesn't on the D region. The D region that codes for amino acids only codes for 3 of them and is therefore only 9 nucleotides long. And as it turns out, each of the flanking sequences around it is longer than that. So we'll get back to that. Here we focus in on a portion of the kappa light chain gene. So up here we can see the leader variable sequences, followed by the recombination signal sequence. We'll see here we have the gray region, which is going to be the heptamer, the lavender region, which will be a one-turn base sequence, and the orange-red region, which will be the AT-rich. So, and downstream below that, we have, again, another AT-rich, a 2-turn sequence, and a heptamer followed by the yellow J region. And so when we're done, we're going to put together the V with the J, and let's look a little bit in more detail about what these really represent. All right, so what you will get in any recombination signal sequence is next to the region to be attached, you will have a 7-base heptamer. That's what heptamer means, and we'll see that it's palindromic when we look at the specific nucleotides. So here at the end of the V, the beginning of the J, are the 2 heptamers, and eventually they're going to actually contribute some nucleotides into the final product. Notice that the next thing that comes is either a 1-turn or a 2-turn sequence. As it turns out, the way these things are set up, you must put a 1-turn with a 2-turn, and that kind of prevents you from sticking 2 V's together or 2 J's together, because those 2 sequences are important in lining up this whole procedure and making sure you're putting the right things together. The last sequence, or the sequence in the middle, is an AT-rich nonomer, which is also important in recognizing the multi-enzyme complex that puts these 2 guys together ultimately. So when we're putting 2 sequences together, we have to put a 1-turn with a 2-turn, although if you look at the lambda, the 2 turns follow the variables and the 1-turn precedes the J's. But the principle is the same whether you're looking at either light or the heavy chain gene. So now let's go in and get a further closer look. And here we can see the nucleotides. If you'll recall, I said that this heptamer region of 7 bases was palindromic. And if you look at this thing, it reads the same way front and back. So I could read CACAGTG, or I could go the other way around and go CACATGTG. on the backwards one. And that's true on the other side as well, and you can see they're kind of flip sides of each other. The one-turn sequence has 12 bases of, any old thing, that's why it's an X, and the two-turn has 23. So what does that mean? What it means is if I start out one place on the DNA and I count off 12 bases, I will find that I have made a full lap around the molecule. That is, I've made one turn. And I have this illustrated on one of the supplementary videos where I've got a 3D DNA model and I can show you what happens when you count around it. Likewise, when you look at the two-turn sequence, I think you're probably not surprised to know that if you count 23 bases, you will make 2 laps around the DNA. And this 23 base one I'm doing in this sort of deep bluish purple, and I'm doing the 12 base one in a lighter lavender so that you can tell them apart by color. The AT rich regions are, again, pretty much the same. Notice it's A's on one side and T's on the other. And this is a nonomer, or 9 bases, okay? So, running from the region to be pasted together, we have a palindrome, a turn sequence, and an AT, and then going the other way, facing it is another AT, a turn sequence, and palindrome. And those sequences will be read in order to essentially paste the respective V's and J's together. Now, if we go back and take another look at the signals, we'll see again we have a bit more complication with the heavy. The first thing we're going to do is put together the D and the J, but notice there's no way on this one at least to say, for example, put 2 D's together, or likewise we can't skip the D and put the V and the H together because that would put 2 turn signals together, and that's a no-no. So we have these sequences. They're going to help the DNA attach to and be moved around and chemically rearranged by an enzyme complex called VDJ recombinase. And we're going to look at the exact mechanism of this in the next sequence.